introduce Dr. Mazda Janab, who is a scientist at the International Agency for Research in Cancer in Lyon and has a long history in examining mechanisms leading to cancer, and will focus today on obesity specifically. Mazda, please.
Good morning. Thank you very much to the WCRF and to the organizers for their very kind invitation to me. I also declare that I have no conflicts of interest whatsoever pertaining to this talk. Over the past previous decades and many years, there's been a lot of insightful and very in-depth studies on mechanisms underlying diet and lifestyle, environmental exposures, and cancer development. And my intent in a basic 15-minute talk is not to summarize, is not to go into any detail of these, but just to summarize the basic understanding. And it comes down to something of this respect, in the sense that unhealthy dietary and lifestyle patterns, the energy imbalance, obesity, and diabetes that they may entail has metabolic consequences, such as hyperinsulinemia, hyperglycemia, hypertriglyceridemia, and higher levels of growth factors. And then these cause a growth-promoting environment. Another hypothesis is to do with hormonal factors, that there are disruptions of hormonal metabolism, steroid hormones, and hormone receptor signaling, and so on. And this causes increased cell proliferation and decreased apoptosis. And a third hypothesis has to do with altered synthesis and production of adipokines and cytokines, leading eventually to increased levels of inflammation. Now, together, this inflammatory and proliferative environment over the long term is growth promoting, and the underlying understanding is that this leads to increased cancer development. One way to understand this concept of metabolic dysfunction is to look at metabolic syndrome, which is a cluster of metabolic abnormalities throughout the body. In this slide, I put up the results from a study based on the EPIC cohort, a large prospective European cohort, where the metabolic syndrome score was assessed by its three definitions, and the risk for development of colon and rectal cancers was also assessed. And you can see in the column in orange that the risk for colon cancer is increased with the presence of metabolic syndrome. For rectal cancer, the picture is less clear and not statistically significant. And by this slide, I intend to show two things. First, that metabolic dysfunction does appear to be mechanistically involved in cancer development, but secondly, that not all cancer sites are equal in terms of the impact of these underlying mechanisms in cancer development. That is exemplified here on this slide, where we show metabolic dysfunction with and without overweight, where we compared normal weight individuals or overweight individuals who are metabolically healthy versus normal weight or overweight individuals who are metabolically unhealthy. And we observed that with metabolically unhealthy individuals, irrespective of being overweight or normal weight, the risk for colorectal cancer is increased significantly. It's interesting to note that there is a large overlap between the risk factors for cancer development, the risk factors for other obesity-related conditions such as diabetes or diabetic kidney disease, in the sense of inflammation, the role of the colonic microbiota, vitamin D, and variety of dietary and lifestyle factors. And this suggests to us that these exposures, the mechanisms, and the disease endpoints do overlap and are linked in some way mechanistically. Of course, there's a lot of questions that we don't know about these aspects. Does the association hold in all populations? Is there a sex-specific difference? Is there a molecular signature for obesity-related cancers or obesity-related disease that one can decipher? And are there molecular targets that then can be modulated or targeted for stopping these mechanistic processes? And importantly, are there gene-environment interactions and genetic predispositions, which is information that we know very little about. Another mechanism that I spoke about earlier was inflammation. And on this slide, I show results from the EPIC prospective cohort where C-reactive protein, a sensitive but non-specific biomarker of systemic inflammation, was analyzed in association with risk of developing colon, rectum, or the combination colorectal cancer. And you can see in the top part of the slide of the graph with the association with colon cancer is quite clear. There's an increased risk with increasing levels of circulating C-reactive protein. For rectal cancer, however, the observation is a bit muddy and unclear. And by this, again, I want to demonstrate the role of inflammation in cancer development, but also that the mechanism is not necessarily fitting to every cancer site. Of course, one can think that inflammation is related to cancer development, therefore stopping inflammation would be related to cancer prevention. And indeed, this question has been analyzed recently in the multi-ethnic cohort study, another large cohort study in the United States, where individuals taking NSAIDs, whether it's aspirin or other kinds of NSAIDs, were assessed, and their risk for developing colorectal cancer was assessed. And on the left-hand side of the table, you can see the risk in men, and you can see with greater consumption of NSAIDs, the risk for developing colorectal cancer is significantly reduced. The same, however, is not true for women, shown on the right-hand side of the table, where the outcome was largely null. And by showing this slide, my intent is to point out the sex-specific potential of some of these mechanisms, that the effect in men and women may be different based on the cancer site and the exposure and the mechanism. If we look at the molecular level at the different details of the mechanisms, it's interesting that they line up almost very nicely with the known and emerging hallmarks of cancer development. And this shows us that going from exposures to cancer development through these mechanisms, those exposures being obesity, lack of physical activity, or unhealthy dietary patterns, can have a meaningful explanation through these mechanisms. There are a lot of emerging mechanisms that we are beginning to understand, and we require more in-depth research about. And this slide shows metabolomic profiling for understanding the obesity and cancer risk. Now, we know that higher body mass index and obesity is associated with a degree of metabolic dysfunction in the sense of changes and differences in circulating levels of a number of compounds, such as acylcarnitines, amino acids, phosphatidylcholines, biogenic amines, and others. And so to assess the association between these metabolites and colorectal cancer development, a joint project between IARC and the Imperial College of London looked at targeted metabolomics and the risk of colorectal cancer development, analyzing the metabolomics in pre-diagnostically collected blood samples. And it's interesting to see the metabolites, both positive and inverse associations. However, only 3 metabolites were statistically significantly associated with, inversely associated with colorectal cancer risk: histidine, equol, and a phosphatidylcholine. And it's interesting that these 3 metabolites tend to derive from colonic microbiome metabolism, which suggests that the microbiome may have a role in colorectal cancer development from this novel analysis. In my group at the IARC, we also looked at metabolomic profiling for looking at hepatocellular carcinoma, looking at the underlying mechanisms of this cancer, which has a very strong link to diabetes and to obesity, but also other etiologies, specifically viral, but also alcohol abuse. And so it's an interesting cancer to look at in this sense, to be able to dissect apart the different etiologies of the cancer development process. And in this study, based on a prospective cohort, the EPIC cohort, we looked at 4 different LC-MS metabolomic methods to get a wide range, a large spectrum of metabolites in different metabolic pathways. And the table that you see on this slide shows the top 16 hits that we had from the metabolomic analysis that we could annotate. And you see both inverse associations, shown in green, and positive associations with hepatocellular carcinoma for these metabolites, shown in red. And when you look at the speculated role of the metabolites in HCC development, it's very interesting to note that they come from a variety of different pathways. For example, retinol has a role in immune function and cell growth. There is components of insulin sensitivity or insulin resistance. There are dietary components such as vitamin E metabolism, markers of liver function, and also markers of liver damage positively associated with HCC. Amino acid from meat protein. There are markers of microbiome functionality within the colon, food additives, particularly from sugary drinks, carcinogenic compounds, bile acids, which is another emerging underlying mechanism for cancer development, not just colorectal, but for liver cancer as well here, and even exposure to methylating agents and DNA damage. So untargeted metabolomic analyses can provide us insight into multiple mechanisms, multiple pathways, and multiple etiologies within the cancer development processes and its link to obesity. Another emerging mechanism is related to the microbiome is the integrity of the gut barrier. We know that the microbiome may be modulated by obesity and overweight, and the gut barrier may be broken down, allowing leakage of carcinogenic compounds into the systemic circulation. In this study, we analyzed serum endotoxin exposures as a marker of the functionality of the gut barrier. And it's quite interesting, on the top row, the results for men, we see a statistically significant increased risk for colorectal cancer with decreased gut barrier function. Whereas in women, the association, although not statistically significant, is almost the opposite. And this highlights not only the potential of this mechanism as having a role in colorectal cancer development, but also, again, the sex-specific nature of some of the observations that we are seeing. I come back now to some of the discussions that Professor Leitzman had in his presentation, the association of obesity with cancer development. And it's important to note from some of the earlier data that I presented, it may be perceived that obesity is something that is measured at one point in time. And in all of our studies, it is. But the development of obesity, the link of obesity to cancer development, and the underlying mechanisms are long-term processes, taking many years. And so in this study, based on the Chances Consortium, which is an EU-funded program, the duration of obesity, overweight duration per 10 years, and the overweight years in units was assessed in a number of different cohorts and different populations with the risk of developing obesity-related cancers. And you can see that with increasing duration of obesity, the risk for cancer development is increased dramatically and significantly. And this suggests that there may be windows for cancer prevention in preventing obesity and even losing weight. And that leads me to my next slide, which has to do with weight loss intervention. This INTERCEPT study is a joint study between Mark Gunter, who was originally supposed to give this talk, and colleagues at the University College of London, where a number of obese subjects were enrolled and biological samples collected before and after either intensive weight loss, which is a very low-calorie diet, or general dietary advice for weight loss. And a number of molecular pathways were looked at. This slide shows the very preliminary results from the INTERCEPT study, where after 18 weeks of the intervention and a mean weight loss of about 20% in some of the subjects, large and significant decreases in growth factors, in insulin, and inflammatory factors were observed pre-intervention compared to the post-intervention period. And even down to the cellular level, with these two pictures showing the colonic crypts and the level of cellular proliferation in the crypts, you see before and after weight loss. After weight loss, there's a dramatic decrease in cell proliferation. Also, gene expression was compared pre and post weight loss. And you can see in this slide general changes with weight loss across the intervention period in gene expression, particularly genes pertaining to obesity and cancer processes. So weight loss can have dramatic impact on some of the underlying mechanisms for cancer development. I go now full circle again to the WCRF guidelines for cancer prevention, and this slide shows results from a study that we did on, in collaboration with Imperial College of London, on adherence to the WCRF 2007 guidelines and reduced risk of cancer development at a large number of cancer sites and in total cancers. And you can see that with simple advice for cancer prevention, the risk for cancer development is reduced at a large number of anatomical subsites. And this suggests that simple advice, simple or even simple policy factors leading to the next presentation may be quite effective, but we still need to have a good understanding of the mechanistic underpinnings. So in summary, obesity and physical activity are risk factors for a number of cancers that I listed, many of which are becoming increasingly common. Nevertheless, we have a lot of unanswered questions around the causality, the specifics of the mechanisms, heterogeneity across cancer subtypes, and sex-specific effects. There is ample evidence to implicate hyperinsulinemia, hormonal factors, and inflammatory factors as part of the biological mechanisms for cancer development. Yet there are some vagueness in these associations that we still need to assess. And so future and other studies should look at very advanced omics methods, such as metabolomics data that I put up, and try to look at different study designs to assess the underlying mechanisms. I'd like to thank and acknowledge the many collaborators underlying this data, and again, the WCRF for their invitation, and you for your attention. Thank you.
Yes, thank you very much, Master, for the presentation. Now we have enough time to take a few questions. Anyone interested in more information about the mechanism? Maybe I should start with the question. The data showing the association between weight development over the lifetime and cancer risk was quite intriguing. Do we have any information regarding mechanism, in the sense that these mechanisms would run in parallel to those from which we know already that they are associated with obesity measured at one time point in cancer risk, or is there additional mechanism work to be done and evidence from which we could learn?
Your question is a very important one, Michael, in the sense that our assumption is that throughout the duration of development of obesity, that the mechanisms are likely to be the same, but that's our assumption. There's very little work on the impact of obesity throughout the life course, and whether the mechanisms that pertain to the link between obesity and cancer development at different time points in life are the same or vary. We may speculate that they may vary. For example, the impact on gene expression or metabolic dysfunction. We know that metabolic profiles vary by age, and so these aspects certainly pertain to these associations. The problem is that we have very little data on that topic and very few research infrastructures that can address these questions.
Yes, please.
Thank you very much, Dr. Janab. It was a very nice presentation. I have a point. You showed that the relationship between obesity and cancer in different studies And I was looking at carefully that in the EPIC study in Spain, the risk is the half, whereas in Denmark and the other country, using the same system because they are EPIC country, there is a very big difference. How do you comment this?
There is a point in what you say, in the sense that there are likely to be heterogeneity amongst populations with different exposures. So different populations in different regions of the world may not have the same associations, and this is one potential example. There's, of course, the concept of adjusting for confounding, and our studies were fully adjusted for confounding, but one can never address that concept of residual confounding, which may pertain in some observations. There is a lot of work to be done to understand these associations in different populations, and this is a good example of the need for that work.
Any further questions? Okay. That is not the case.